Hello and welcome to Only Sign No File. Don't forget to like, comment, share, and subscribe. In this video, I'll be talking about FACS, that is fluorescent activated cell sorter. Now, this is a specialized type of flow cytometry technique. I've already covered the basics about flow cytometry in the previous video, so if you haven't watched it yet, I'll be adding the link in the description. Now, FACS is used to sort different types of cells, and it is the quickest and the most effective way. It is based on the principle of forward scatter and side scatter, as I already mentioned in the previous video, and along with those two, fluorescent signal data. How? I'll be talking about that in a minute. Now, suppose you have a mixture of cells. Now, firstly, you want to identify a certain type of cell or you want to sort them. That is, this is a specific cell which you want to isolate or sort from the other type of cells present in the mixture. Now, example, this is a certain type of protein which is expressed. What you do is you select a particular antibody which binds to this protein, and this antibody is tagged to a fluorescent label. Now, in this case, it's a green fluorescent tag. So what you do is now you take this antibody mixture, and that is antibody along with the fluorescent tag in this particular mixture. Now the cells are going to be attached to these antibodies which have these specific proteins. Now the next step is you inject this in the flow cytometer. What it does is, as I already mentioned, this is a particular representation of an instrument wherein this nozzle allows only one cell at a time to pass through it. Now what next is done is a laser beam is bombarded onto this particular cell, and then there is going to be forward scatter, side scatter, as well as some fluorescence is going to be there. Now, as I already mentioned, forward scatter is detected by the detector. There are side scatters at 90 degrees to this particular setup. And then there is also another detector which measures fluorescence emitted by this particular isotope. That is, some laser beam is bombarded, the electrons get excited. Now, while coming to its ground state, they emit excess energy, which is in the form of fluorescence detected by the detector. Now, all these detectors are then connected to the photomultiplier, which amplifies the signal, and then ultimately to the computer, which analyzes the results and interprets it. This is an interpretation of how it looks like. As I mentioned, there is a cell suspension, drop by drop. Now, this is called as a drop, which passes through a laser beam, which is bombarded. There are forward scatter detectors, side scatter, as well as various other detectors which measures fluorescence, and this is connected to the photomultiplier tube. And how do you exactly now sort the cells? Now this is how, as I already mentioned, the laser, forward scatter, side scatter, and fluorescence detected by the detector. Now what next at this point you do is you impose an electric charge onto every molecule. Suppose now you already have the flow cytometry data. Suppose that particular protein of interest which was expressed on that cell, you want that cell to be collected. Suppose that is a green cell. Now, what you actually do is you impose a certain charge, whichever charge you want, on this particular cell and the other charge on the remaining cells. So what happens is this particular charge, if it is positively charged, you impose it to be positive charge, goes to this deflection plates or electrodes. Now this positive charge is going, the cells are going to be here towards the negative electrodes, as I can say, and the other cells towards the positive cells, and some of them are differently charged. So based on these electrodes or deflection plates, cells are going to be sorted. What you do is you decide that I want this particular cell to be positive or negatively charged, and then now this machine, on the basis of the previous information, imposes the charge you want and then sorts it into different cubes. Now, this is how exactly a flow cytometry or fluorescence activated cell sorter works. So this was all about it. If you like this video, don't forget to like, comment, share, and subscribe.